but then difficulties with technology. So we won't do that. All right. So last time we were starting our discussion of the four forces of evolutionary change, and we had really just gotten into the first one, which was mutation, talking about genetics and mutation. Any questions about that, or is that all coming back to you? Okay, good. Then the next thing we were about to start was random genetic drift, and we didn't really have time to get into it much, so I just kind of cut us off. But random genetic drift is just changes in allele frequency due to random mortality events. The death doesn't have anything to do with the phenotype of the individual. It just has to do with you happen to be in the wrong place at the wrong time. So imagine taking a coin. Avery, help me out here for a minute. Here's a quarter. Flip it. Head wins. Head lives. Tails dies. Heads lives. Well, wait. Stop. No, never mind. How many individuals do we have here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. We have 12 individuals, which means we have how many alleles? 12 individuals, which means we have 24 alleles total. How many of these alleles are the big A allele? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 out of 24 of the alleles are the big A allele. 12 out of 24 are the little a allele. This is P in Hardy-Weinberg equilibrium parlance. So we have allele frequencies that are at 50%. Okay, now flip the coins. You said this individual lived. Flip the coin on this individual. Dead. This individual. Dead. Alive or dead? Lives, okay. Okay, Elizabeth can flip. Heads is dead. No, heads is alive, sorry. Heads or tails? Huh? Heads. You guys are terrible at flipping coins. Heads, it lives. What one were we on? The heterozygote? Heads lives. I'll just flip the coin. Heads lives. Tails dies. Heads lives. Tails dies. And heads lives. So, just through the random flip of the coin, we now have 1, 2, 3, 4, 5, 6, 7, 8 individuals. Which means we will have how many alleles total? How many of those are the big A allele? One, 2, 3, 4, 5, 6. 6 out of 16 are the big A allele, which basically means that 10 out of 16 are the little a allele. So this is 3/8. This is the least obvious form of selection, because this is a situation where the phenotype doesn't change, the average phenotype doesn't change, but the variance still decreases. How can we have the mean not changing, but the variance getting smaller? Selection against extreme phenotypes. Stabilizing selection is when the average phenotype doesn't change and the variance decreases. This is stabilizing. So these are the three illustrated. Directional, stabilizing, disruptive. And in all of these cases, it's just a difference in terms of which portion of the beginning population is being selected against. Now, I've shown you these traits as normal curves. Where do we get normally distributed trait distributions from? I jumped right ahead to this as if you already knew this, and I made that assumption because you've seen this in another class. What kinds of things result? What's happening underlying this trait distribution? How did we get this? If this is a distribution of short individuals to tall individuals, very few short individuals, lots of tall individuals. Like, where did that distribution come from?
Random sampling of the population?
Hmm?
Is that random sampling of the population?
Nope. It has to do with genetics. Which one of you wants to do this? Somebody do this cross. If it's heads, it's Elizabeth. If it's tails, it's Avery. It's Avery. Give us the F1 generation and walk us through what you're doing as you do it. The first thing you do is you make gametes. Yep, there you go. Well, those are both the same. Those are both the same gametes. Yeah, but that's not what you have there. You just have what you have there. Don't be putting stuff on the board that I didn't put on the board. So this individual can only make that type of gamete. What kind of gamete does this individual make? That type of gamete. So what are the offspring going to look like? Okay. Now cross those with one another. Allow those to interbreed. Or self, for that matter. You haven't quite given yourself enough room, have you? Basic Mendelian genetics coming back. Gotta move this closer so people at home can see. If you lecture long enough, you know to give yourself room for things.
Well, I don't know, like
Oh yeah, yeah.
Like, so
Hmm?
off of what you were asking,
Yeah.
like, all of them are tall, or like you have a more tall population because like tall is the dominant phenotype. The majority of them like, have the.
But that's actually not the case. You have just as many tall individuals as you have
Oh, I
short
thought you were
individuals.
saying you had a lot of tall. My bad. I
Nope, nope, nope,
misunderstood
nope.
you.
Okay. Thanks, Avery. So, do you guys remember what a quantitative trait is in genetics? Quantitative trait is a trait that is determined by multiple genes, and each dominant allele contributes a little bit to the phenotype. So what matters isn't the genotype itself. What matters is how many dominant alleles you have. And so in this case, I want to remind you of a couple of things. I want to remind you of the symmetry of the Punnett square when you do it this way. Notice that this genotype is the same as this genotype. Notice that this genotype is the same as this genotype. Notice that this genotype is kind of opposites of one another. Homozygous dominant for the first, homozygous recessive for the first, homozygous recessive for the second, homozygous dominant for the second. These are all the same. This is the same as this. This is the same as this. And then this guy's there. But for quantitative genetics, once again, we don't really care about the genotype. What matters is this individual has 4 alleles that are dominant. This individual and this individual have 3 alleles that are dominant. This individual has 3. This individual has 2, 2, 2, and 2. This individual has 2. This individual has 3, 2, 1, 1, 0, 1, 1. 4, 3, 3, 2, 4, 3, 3, 2. 3, 2, 2, 1, 3, 2, 2, 1. 3, 2, 2, 1, 3, 2, 2, 1, 2, 1, 1, 0, 2, 1, 1, 0. So there's this symmetry, but what matters from the standpoint of quantitative genetics is if we look at the distribution, one individual has 1, one individual has 4. 1, 2, 3, 4. Whoa, 1, 2, 3, 4. 4 individuals have 1. 1, 2, 3, 4. 4 individuals have 3. And the intermediate phenotype, 1, 2, 3, 4, 5, 6, has 6. What is that beginning to look like? It's beginning to look like a normal distribution. If height is a characteristic that is governed by multiple genes, and each dominant allele of those genes contributes a little bit to height, you will get a distribution that looks normal with some individuals that are very tall, some individuals that are very short, and a bunch of individuals clustered around the average phenotype. That distribution up there is generated from doing this cross, but that's a little much to ask Avery to do in class because we get 64 different genotypes. Some of them are going to be repetitive, but this is just a smoother version of that thing that we can do with 2 genes contributing to the trait. So whenever we see curves like this, I want you to always remember those curves are underlain by simple Mendelian genetics. Don't ever forget that. They're not magical or anything else. They're just governed by the same genetics that you learned, at the very beginnings of your genetics class. So when we see plots like this, so this is your conscious and unconscious selection from lab a couple of weeks ago. This is one form of selection. You plant larger seeds before you plant smaller seeds, As a result, if you get to the end of your field before you get all these small seeds planted, those small seeds are selected against, meaning that only the larger seeds were planted. And if there's a genetic basis for large seededness in plants, over time, your plants are going to get bigger in terms of their seed size over time. This can either proceed fast when the selection is strong, or it can proceed more slowly when the selection is not as strong. So we can actually project into the future what happens with the evolutionary trajectory of a trait by using Fisher's Breeder's equation. And I'm pretty sure you don't remember this, so I'm just going to give it to you. Basically, delta Z is the change in the phenotype, the change in the average phenotype. Sometimes it's referred to as the evolutionary response to selection. So another way of writing delta Z is with a capital R. So, and actually that's the more intuitive way of notating it. R, or delta Z, is the evolutionary response to selection. So we could also notate this delta Z, and that's what's on your screen. H squared is heritability. It's the measure of the degree to which the trait is underlain by additive genetic variance, so to the degree to which it is determined by underlying genetics. And S is the strength of selection. So do you guys remember how we determine heritability for these types of quantitative traits at all? And you go back and dredge up all this stuff from your first year. We do this with regression. So in this case, this is an actual data set from a first year evolutionary ecology class that I had. That particular year, we had 64 students in the class, which is just shocking to me. We had so many students. But Over here, now it's just over here. What is the heritability of this population? 0.6615, way lower than 0.9165. Selection gets rid of variation. Heritability is just a measure of the amount of variation for a trait in a population. So one of the things that happens over time is directional selection reduces the variability of the trait. When we were going through these figures here, what does directional selection do? It shifts the mean and it decreases the variance. Directional selection shifts the mean and it decreases the variance. Okay? It's good when the math works out. Now, unfortunately, life can be more complicated than this. Not all characteristics evolve by themselves. In some cases, traits are correlated with one another. So, it's an example that is not true if you look at it more closely, but I'll use it because it's intuitively obvious. You guys familiar with track and field? If you're a good sprinter, what are you generally not good at? You're not good at distance running. And if you're a good distance runner, what are you not particularly good at? You're not good at sprinting. And so endurance might be negatively correlated with sprinting ability because something about your muscles, the kinds of muscle fibers that you have, et cetera, make you good at one thing, and as a result of that, you're not very good at another thing. And so you might think of a trade-off individuals who have a lot of endurance typically end up being slow. Individuals who are really fast tend to have lower levels of endurance. And so you have this negative correlation between speed and endurance. This actually turns out to be a physiological trade-off in terms of the kinds of muscle fibers that you have, the relative abundance of those, et cetera, et cetera. And so we can have these two traits, speed and endurance, that are negatively correlated with one another. And we can measure this, and we can measure this by looking at the parental speed and look at average offspring endurance. So the same kind of parent-offspring regression we used to measure heritability, we can also use to measure genetic covariance. The degree to which two traits covary, and that covariance is underlain by a genetic basis. And so if we're interested in following two traits, the evolution of sprint speed and the evolution of endurance, we need a couple of things. We need to know what the heritability is of sprint speed and endurance. If this is sprint speed, this would be the heritability of sprint speed. This would be the heritability of endurance. And these two things would be the genetic covariance between sprint speed and endurance. This is the degree of directional selection acting on sprint speed, and this is the degree of selection acting on endurance. So we can actually look at these two characteristics evolving in tandem with one another. If we want to know what the evolutionary response of speed is, we simply take the heritability of speed times the amount of selection acting on speed, and we add that to the genetic covariance between speed and endurance multiplied times the selection acting on endurance. So speed is affected by selection acting directly on speed, but also the effect of endurance acting on speed, because these two things happen to be negatively correlated with one another. In a similar way, if we want to know the evolutionary response for endurance, we get the heritability for endurance multiplied times the direct selection acting on endurance, but then we add to that the genetic covariance between speed and endurance times selection acting on speed. So speed can affect endurance by acting through this negative correlation. You look quizzical.
I'm just trying to think what to write down so I remember this. So I do understand.
Okay. The way to prepare for this on a test or in life is to actually work an example. So let's do that. So you may or may not remember this example. This is some work from Denny and Butch Brody. He worked on snakes in Oregon. In Oregon, there is a species of snake called Thamnophis Ordinoides. This is a type of garter snake. Occurs in the Pacific Northwest. And he was interested in this snake because within a given field, within a given population, you will find some snakes like this that are very, very striped looking, and you will find other snakes like this that are more checkered in their color pattern. So there's one character, and you can measure scales and coloration and things on these, and you can build a quantitative measure of stripedness. And so he did that, and so he created an index, a quantitative index for stripedness. That went from a negative value to a positive value. Individuals with a negative stripedness were checkered. Individuals with a positive stripedness were striped. And you find that this is roughly normally distributed in the population. Some snakes are checkered, some snakes are striped, and some snakes are in the middle. He also was interested in their performance, their sprint speed, and their endurance. And what he found, as he was racing these, he would race the snakes around in a circular racetrack and measure how many trips they made it around before they finally got tired out. But one of the things that he noticed is that when they got tired out, as they got tired out, some of them would continue to just run in a straight line. Some of them tended to reverse themselves, and then they would stop. And so he created an index called reversals, with, once again, going from a negative to a positive. Low values for reversals were snakes that ran in a straight line, slithered in a straight line. Positive reversals means that they reverse themselves a lot during any given trial. And so he has this morphological character, and he has this behavioral character. And you can get a bunch of snakes, measure their stripe, get a bunch of those same snakes and measure their reversals. And if you have a mind to do it, you could measure, if you have parents and offspring, you can measure the average parental stripe value and the average reversal of parents. And you could compare those to the offspring. What do you expect the correlation between these things is? Huh? Positive correlation, which means what in terms of the Breeder's equation? Heritability. Heritability is positive. Same thing here. Heritability is positive. When he looked at the correlation between offspring reversals and parental stripe, guess what he found? I think this is Butch Brody racing snakes down a racetrack. The elder Butch. He found that checkered snakes tend to reverse themselves a lot. Striped snakes tend to not reverse themselves a lot. He found a negative correlation between these two traits. Now think about the vertebrate eye. We've been talking in lab about peculiarities of our senses. So we talked in lab about the sensory bias of our fingers as we draw seeds out of a bag. The sensory bias that forces us, even without us thinking about it, to draw large seeds out of the bag before we draw And what we see when you look at selection, this is an adaptive landscape of this trait. We see that striped snakes running in a straight line have the highest fitness. Checkered snakes that reverse themselves a lot. This is reversals. This is stripe. So negative stripe is checkered. High reversals is reversing a lot. Checkered snakes that reverse themselves a lot have high survivorship. Striped snakes that go in a straight line have high survivorship. Checkered snakes that go in a straight line have lower survival than checkered snakes that reverse themselves. And the worst snakes are striped snakes that reverse themselves. So this is fitness consequences of these combinations of phenotype that result, presumably, in this genetic correlation. So if there's no genetic correlation, what would be the rate and direction of evolutionary change? So these are actually figures from Brody's work. And in this case, reversals is on top, stripe is on the bottom. Heritability of reversals is relatively low. Heritability of stripe is relatively high. Selection against reversals is relatively weak. Selection against stripe is considerably stronger. And in this case, there is no genetic correlation between these. So the way you figure out what's going to happen with the evolution in the system, if this does not exist, is just heritability times selection on reversals, heritability times selection on reversals, this times that, plus this times that, which ends up being 0. If you want to know how stripe is going to behave, it's this times that plus this times that. This is reversals acting on stripe through the genetic correlation. And when you figure that out, this is 0.002, this is 0.0721. These are both 0 because there's no genetic correlation. And so this is the direction of change of evolution. You can expect a little bit of change in reversals and more change in stripedness. Stripedness changes more for 2 reasons. One, it's being selected on more strongly, and it has a higher heritability. Less selection with a lower heritability results in less change. But now what happens if we put in the negative genetic correlation? Oh, so this is what's happening. That's all that happens. You have the effect of direct selection acting on reversals plus the direct effect of selection acting on stripe. But if you include the negative correlation, it turns out the negative correlation was quite high in this particular system. So now we have the direct effect of selection on reversals, 0.245 times 0.008, right there, added to a negative 0.751 times 0.102, a high genetic correlation coupled with a high degree of selection on stripedness. For reversals, we have heritability of stripedness. Sorry, did I say reversals or stripedness? For stripedness, we have high heritability of stripedness times selection on stripedness, but then we have a high negative correlation being acted on through selection on stripe as a result of selection on reversals. And when we do the math, what we find is that the directional selection, the direct selection acting on these 2 traits, hasn't changed at all, but now from this we subtract selection due to the other trait. And we subtract selection due to the other trait. And so what we see is now rather than reversals increasing slightly, they're actually decreasing because reversals is being drugged down by this negative correlation and selection on stripe. Stripe continues to increase, but it's not increasing as fast as it was before because it's being slowed down as a result of this negative genetic correlation due to reversals that then affects stripe. So in effect, what you have here is if you start here, you have the effect of direct selection acting on reversal and stripe, but then you have the correlated effect of reversals acting on stripe and the correlated effect of stripe acting on reversals. Sorry, this is the effect of stripe acting on reversals. This is the effect of reversals acting on stripe. So what you do is you see, rather than these two things increasing, you see one actually decreasing quite a lot, and the other one not increasing nearly as much as it would. You see reversals decreasing a lot from where it was, and you see stripe increasing, but not as much as it would have because of the counteracting effect of selection. And so this ends up driving populations towards one of these two peaks, where you have striped snakes going in a straight line, checkered snakes reversing themselves a lot. So this is multivariate evolution. And the effect of these negative correlations is that they either cancel out or slow down evolution because there's this negative correlation between traits. We can also have positive correlations, but those will do just the opposite. They'll speed up the rate of evolution. And so when you think about an organism out there in the world doing its thing, you have to think of it not as a bunch of atomized little traits that you can look at in isolation from one another. You have to think of it as a whole integrated organism. As a result, you might have multiple traits that are all evolving in concert with one another. And so while you might observe selection acting on one particular trait in a particular way, it may not evolve that way because it may be correlated with something else that is under stronger selection. And so you have to start to think about these organisms that are out there on a landscape behaving as a combination of lots of traits that are all put together in this one single individual. So the direction and rate of evolution is ultimately determined by the shape of the adaptive landscape, who survives and who doesn't. But it's also determined by this genetic architecture of the traits that are involved in evolution. If you have these negative genetic correlations, then evolution in general is going to either be slower than it would be otherwise, or it may even go in directions that you don't expect if you just look at the trait by itself. And this is one of the challenges of dealing with complex traits like behavior. This leads to constraints. So this is one of the constraints. You can't be striped and also reverse yourself a lot. That's a bad combination. So colloquially, we think of constraints as, for every advantage, there is a disadvantage. You can't have your cake and eat it too. Superman has X-ray vision. He can fly. He's super strong, but can't stand kryptonite. What are some constraints that you guys deal with? I can't have fun on Thursday night and do what Friday at 12:30? This Friday at 12:30. Yeah, you probably can't go out and have a lot of fun on Thursday night and then come in here on Friday and do well on the test. We are constrained by time. Any time that I spend preparing for class is time that I can't be doing something else. Any time that you spend going to the movie is time that you can't spend, practicing your sport or studying for exams, etc., etc. So colloquial constraint is just not being able to do it all and do it all well. We have this myth in the United States that you can do it all. Well, you can't really do it all. You have to pick and choose. what you're going to spend your time on, because if you try and spend your time on everything equally, you end up doing none of it very well. So time devoted to one activity is time that you cannot devote to a second activity. Unless you happen to be in applied stats where you're surfing Facebook at the same time that you're supposed to be listening to applied stats lecture. Not you, somebody else in the class. No, it wasn't you, it was somebody else. And as soon as I said that, I made that Facebook crack, he turned his Facebook off. But there are evolutionary constraints, and those are the kinds of constraints that I'm concerned with. And these can be morphological constraints. So a morphological constraint might be well, what would be a good morphological constraint? I should have one ready at the waiting because I knew that this was coming. Oh, morphological constraint. If you are a giraffe, the long neck that you have enables you to get to tall vegetation very easily, but it also makes drinking something of a challenge. Um, we have constraints in humans. This is human birth weight data. So this is babies, the weight of babies at birth, and the probability of survival. And if you're an average size baby at birth, this is from 1951, so this was back in the days before C-sections were common. If you are an average size baby, you do fine in terms of probability of survival. If you're a premature baby, your probability of survival plummets drastically. But also, if you're too big, your survival goes down as well because you just have trouble passing through the birth canal. Women's birth canal is surrounded by a rigid pelvis that doesn't really flex very much. So if you're too big to go through there, a lot of times you end up, the baby dies and the mom dies back in the day. And so we have, this is a selection surface. It's a univariate selection surface rather than a bivariate selection surface that we saw earlier. But there's lower survival at the high end and much lower survival at the low end. If we were to do this graph today, what would this graph look like? There would be less death overall. This would be less death because preemies still die at a higher rate than a normal birth baby, but we do a much better job now the ability to drive here. So I will let you guys know about that at the time.